There you go. Okay, we're good.
Let me just share my screen real quick.
Oh, and to clarify for the professor that's watching the recording, we're going to do it as a group just because we already were meeting and it's just easier this way. And we're all going to present to each other right here.
Just real quick, I'm just turning off all my ringers so we don't mess up our recording. All right.
I should probably. Are you guys getting my Discord notifications?
Oh, we haven't gotten them. No.
Okay, cool. Just wanted to make sure. Okay. Okay. So my timeline is about the history of forensic pathology. The history starts off with Rudolf Virchow. He was born in Germany in 1982, and he mostly studied cell theory, and he was able to apply pathological process to them, and he was able to emphasize that diseases weren't caused by organs or tissues, but their individual cells. And because of that, he was able to do really impactful studies for forensic pathology. In the year 1959, forensic pathology was first recognized in the United States. In the 50s, the Board of Governors were able to realize that there were sufficient supporting beliefs using pathology, so they were able to certify the forensic pathology and their policies. In the year 1984, DNA finger testing was developed by Sir Alec Jeffreys. DNA fingerprinting is very important in the forensic pathology field because it is able to identify bodies, any blood correlation with other people, and overall how to understand how it affects diseases. Um, Sir Alec Jeffreys was a professor in Merton, Oxford, and he studied biochemistry and genetics. I'm gonna go in the year 1987, Tommy Lee Andrews was the first criminal that was convicted through DNA evidence. On February 24th, 1987, he broke into a Florida woman's home and he burglarized and raped her at knife point. But because there were still semen at the crime scene, they were able to match his blood DNA with it. So he was able to be convicted for it. He served 22 years and he's currently still serving his time, but there's no chance for him of coming out. And ever since then, about 6,000 cases have been solved through DNA testing. In 1996, the DNA testing was certified by the National Academy of Science. This means that the National Research Council was able to interpret DNA evidence to answer key questions like whether there was improper handling of evidence or if there were false incriminations. In 2011, the University of Michigan were able to produced this software that was able to compare hand-drawn facial sketches to mugshots, and they were able to use mugshots from what was already stored in databases. And lastly, here's a video that encaps the overall history of forensic pathology and how it has evolved over the years. Yeah,
Great job.
thank you.
I really like how you have like a lot of images and stuff. I feel like mine isn't gonna be as like visually appealing, you know? All right. So that was Judith's. I will go next. Let's see. There you go. You guys see everything okay?
Yeah, we see it.
All right, cool. So mine's a microbiology timeline. I put my name on there, Eduardo Guzman. Here's my infographic. I had struggled with finding an infographic, and then when I was looking up microbiome infographics, I literally found one that said bacteria infographics. So here it is. It shows. It's kind of a quick summary of everything I'm gonna say, like showing the actual shapes of bacteria and stuff. So starting in 1546, this Italian man whose name I cannot pronounce, just Francastro is the best I can do, suggested that there were organisms around us that might cause disease that we cannot observe. At this point, the current theory was miasma theory or miasma, depends on who you ask. And that theory kind of had to do more to do with like it had something called four humors, and it just kind of, according to scientists at the time, you needed to balance these four different things in your body to stay healthy, and they were like black bile, yellow bile, blood, and phlegm. So somehow by balancing phlegm and blood, you're gonna get over, the Black Death. Um, 1665, Robert Hooke uses an early microscope to look at a piece of cork, and he sees little compartments. He names them cells because they look like jail cells or monk cells, like that monks would live in a monastery. So cells were first observed, and you see the image of his early microscope. 1677, Antony von Leeuwenhoek uses his own form of microscope, which was different than Hooke's, to observe little animals that he wrote about in pond water, as well as the plaque on his teeth, which was like the first known bacteria that we've had like get written about. Louis Pasteur, he disproved spontaneous generation. So it was the prevailing theory that life would just generate from the earth or decaying matter using his swan-necked flasks. So using these kind of sealed off glass jars, he put meat in there, and he was able to prove that maggots don't just appear out of meat, and that like mold and stuff doesn't just appear out of nowhere. It has to be exposed to air. So that kind of gets the ball rolling on thinking that, oh, there might be something responsible for spreading disease or maggots or stuff like that. 1862, birth of germ theory of disease. Louis Pasteur, again, right after, proves that comes up with some of the first evidence to prove modern germ theory, which is, that germs cause illnesses, and there's little creepy crawlies that kind of exist in our world that make us sick. And that was through his continued experiments with the swan-necked flasks. And here is a crash course video that pretty much goes through the timeline up to here. Germ theory gets first proof. Scientist Robert Koch discovers that a small bacteria was behind the human toxin anthrax, which is the first step in putting it into miasma theory. He was able to kind of isolate a certain bacteria that he realized was killing mice, and he was able to do this using his solid media that he came up with. So he was the first time we grew bacteria colony, like groups of bacteria on media, so like a little surface for the first time. And he wrote his famous, they're called postulates. So they're kind of rules or guidelines to determine if a bacteria, like how to determine if a bacteria is responsible for causing an illness, which we still use today. I used it in my class last semester. it still holds up today, which is incredibly beneficial for anyone studying microbiology or even illnesses, epidemiology, stuff like that. Christian Graham invented the gram-negative stain, so that helped kind of put, I don't want to say segregation, so I want to feel like able to separate bacteria, like by how to classify them. That's what I'm trying to say. By using his special new stain that only stained specific cells, and that helped them differentiate. You know, like, oh, this is what's called a gram-negative because it comes back blue versus a gram-positive, which doesn't get affected, which we still use today, and like you see it in modern science textbooks. R.J. Petrie invented the Petri dish. This allowed scientists to now keep their cultures isolated. We still use these today. You think of science in a biology setting, you think of a Petri dish. So this was a really big stepping stone into like where we are today in the field of microbiology. Frederick Griffith witnesses genetic transformation. All that really means is we've got to see how bacteria kind of lives and transfers its genetic information. We didn't know how the hell it did that before, but now we know, and we still use that today in like modifying things and like in the production of, say, diesel, like biodiesel. We use genetic transformation to this day. 1929, first antibiotic. Alexander Fleming rediscovers penicillin. It's an antibiotic that kills certain bacterias, and we use that today to treat bacteria and bacterial infections. You know, your mom, she gets sick, so she gets the penicillina, you know, like it's that kind of thing. And I say rediscovers because we had evidence that the ancient Egyptians had found it first. 1953, Franklin Watson Crick discovered and published the double helix structure of DNA. It was a huge stepping stone. We now knew what DNA was and how it looked, and that eventually lets us start messing with it. 1983, PCR is invented. Polymerase chain reactions procedure. It's a very technical term, but if you think about it, it's a type of procedure that we use to do modern COVID testing, as well as any testing for genetic illness or like a presence of an illness. All it needs is a certain sample of someone's. It could be like a fluid, like a swab of your cheek, or you know, a swab that goes up your nose, and we could test to see if there is a certain gene present in that fluid or material, which was great for identification and for disease treatment. 1995, the first bacterial nucleotide sequence. All that really means is that we are finally able to completely look at a bacteria's genome, which is like finally being able to read a book that you've never been able to read before because you learned the new language. And that lets us do all sorts of cool science things like messing with it or looking into their history. And then she wanted us to include how we're going to join this field. So I put Eduardo Guzman joins a lab. In attempt to further the field I love so much, I want to join a hands-on lab to try and further the research about the field of biology that I love so much. Hopefully, if all goes well, I'm trying to get into a lab this coming fall or spring. And that's my presentation.
Well done, well done.
Thank
You
you.
did a good job.
Thank you.
Geez, nothing like mine looks like that.
Hey, no worries. Mine is all just sadness that I wrote last night.
All right, so here, let me share mine. Okay, can you guys see it? Hello?
Yes.
Okay.
Yeah,
Oh, that
for
looks
some reason I
so
couldn't unmute myself.
cool.
Oh, okay. All right, guys. So, unfortunately, my title site is not showing. I don't know why it's not updating, but basically, so my title slide was there was a little infographic and you know, just my name. So my name is Vikram Kella, and basically my timeline focuses on, like, you know, discoveries in the field of medicine. And so basically, we're gonna start off with in 1796 with the discovery of the smallpox vaccine. And the smallpox vaccine was introduced by Benjamin Waterhorse in 1796. And basically, smallpox was a disease that was contagious and like very deadly, and it killed thousands of human beings for thousands of years. And so basically, the discoverer of the smallpox vaccine started the timeline of medical discovery. There were some discoveries before or prior to this, but we started with the smallpox because this was a serious disease, and this discovery of this really saved the lives of many and for the future as well. Okay, so next we have is in 1843, anesthesia. And basically, anesthesia was discovered by Collins Warren. And the first demonstration was performed in 1843. And John Collins Warren was the school's first dean. And yeah, so basically, anesthesia was is also used in medicine, and typically when there are procedures, surgeries, anesthesia is brought in. Okay, so next, in 1922, the discovery of insulin was introduced by Elliot Jocelyn in 1922, and basically, um, insulin was like a hormone produced to regulate glucose in the bloodstream, and honestly, it is saving so many lives today. In particular, like for an example is like my mother. So she has diabetes as well, and she is actually takes insulin almost daily to control her glucose levels in the bloodstream. And so yeah, and here is a video that you guys could watch whenever you guys get the chance. But it's just further explaining insulin and how it was discovered. All right. So next we have is the iron lung, and the iron lung was discovered in 1927 by an industrial hygienist called Philip Drinker. And basically, um, the iron lung was like a ventilator that allowed polio paralyzed patients the ability to breathe. And honestly, I was just very fascinated by this because it's a lot of lives were lost after they contracted like the polio and became paralyzed. So this thing really allowed the survival rate to get higher, but also for like in this picture how they're still like they're a couple, they're still living life. And I just found it nice, discovery like this in medicine really just made her life better. But yeah, okay, so next, in 1929, so after the introduction of like the iron lung, basically, so the first patient survived from the iron lung was in 1929. And I thought I'd just throw that out there. But okay, so next we have is in 1938, basically, um, the first successful closure of the patient ductus arteriosus, a congenital heart defect of infants, was success by Robert Gross. And basically this had led to an era of corrective heart surgery for children, and it is still being used till today. So this was a major discovery in the field of medicine because now we can work on children and have a better success rate of them surviving from their heart diseases. Next we have is in 1947, it was called the artificial kidney. And basically the artificial kidney was like founded our history of dialysis. And it's just the blood purification and kind of just cleaning out the blood. And it's very critical and crucial even to society today because there are many people that are on dialysis, who have kidney failure. So yeah, so 1947 was when that was discovered. Next we have is platelets. And so in 1960, the development of the means to collect, preserve, and transfuse, and also to collect bleeding was discovered by Dana Ferber. And basically, Ferber kind of discovered like the use of platelets and basically how we collect them, how we preserve them, how we transfuse them. And basically, I just threw out a little definition. That was like a platelet is a small colorless disc shape without a nucleus, and it's found in large numbers in the blood and involves clotting. So this discovery in medicine was also very important as we use it today, and there are platelet donations for some people that have low numbers and can't stop the bleeding. So we have to give them platelet donations and make sure they can control that. Next we have is chemotherapy, and basically this was discovered in 1974 by Mass General dermatologists Thomas Fitzpatrick and John Parrish. And basically, photochemotherapy is a type of ultraviolet radiation treatment that is used to treat severe skin diseases. And as you could see in the picture right there, so there is a skin disease right there, and then that is the effect of chemotherapy, and like the skin is cleared up. And a lot of common diseases we use it till today are like could be like eczema and stuff like that. So we use photochemotherapy to treat those. But yes, another very major discovery that was found in medicine. All right. So next we have is in 1997, the discovery of the P73 gene. And basically this was a novel gene that suppresses tumor, and it's similar to P53 gene, but this was discovered by the HMS cell biology team in 1997. And P73 is found only on one chromosome and acts quite different from its famous relative, which is the P53. And the P53 was also another tumor suppressor gene. And the image below, the image right next to it shows a visual representation of the P73 gene. And yeah. So next we have is actually 2017, which is not too far away from us today. And basically in 2017, the HMS Department of Neurobiology discovered the first explanation to why blood barriers often remain closed. And this is a very important discovery because it could lead to ways to deliver drugs across the barrier of the brain and into the brain to treat disorders such as brain cancer, stroke, and Alzheimer's. And yeah, so definitely open the barriers to treating more brain diseases. And so this kind of wraps things up. But so my topic was, discoveries in like important discoveries in medicine. And basically just a little bit about me is that I'm a second year human biology major, and I fit into this discipline because I am a bio major, right? I am into human biology, I am into medicine, and I find great interest in this. And honestly, in the future, I myself do plan on working in the hospital setting and furthering myself in these like discoveries of medicine, and hopefully getting some hands-on experience with you know kind of using these discoveries, using like dialysis and basically just treating patients. But yeah, and just throw it out there, there was a question where it was like why are you paradigmatic? And basically I just kind of put that there like because as I've noticed, as I've been getting more educated, or as I've been getting more of an education, a lot of my disciplines and ideals are changing because there are a lot of like facts that you believe that aren't really true, but as you get more of an education, you get to figure out why certain things happen or how they happen. So yeah, thank you guys, and that wraps up my presentation.
All right. Yeah,
Great
you
job,
did
man.
good. You did good.
It was really
Okay,
good.
well,
All right, so I'm gonna
I'm gonna
stop
go
sharing.
next, and I guess I didn't get the assignment. Like I didn't understand it, so we'll see how mine goes. Can you see my screen?
Yeah.
All right. So this is the United States government. My name is Jesus Hinojosa. So here's a video crash course. So on YouTube, let me move this real quick. So in 1776, the Continental Congress voted for independence on the 2nd of July. And then 2 days later, a statement outlining the grounds for independence was accepted, which is primarily authored by Thomas Jefferson. Washington got official communication in the form of a letter dated July 6 from John Hancock, the president of the Continental Congress, which included a copy of the Declaration of Independence. And then the Declaration of Independence, the 13 American colonies broke political ties with Britain by signing the Declaration of Independence on July 4th, 1776. The declaration summed out the colonists' motivations by proclaiming independence. The American colonies secured an official alliance with France and French aid in their battle against Britain. And then U.S. political factors and parties began. Parties across during the ratification debate of the 1787 federal constitution, their dislike intensified as the focus shifted from forming a new federal government to its authority. The Federalists wanted centralized control, while the Anti-Federalists wanted state sovereignty. Federalists backed business, while opponents backed farmers. George Washington warned of the dreadful ramifications of party spirit in his farewell speech. Here's an infographic of how our government is separated by three powers: legislative, executive, and judicial. So this is when I became interested in United States politics. This is like literally the first time I was actually amused by it. I didn't know what I wanted to study, everything. I was just like exploring everything. But once it came to like this very moment in my life, that's when like racism, protests, and people wanting to fight for their rights. Um, Throughout the 2016 presidential election, that's when things transpired across my community in Los Angeles. That's also when I witnessed like classroom discussions based on their immigration status and what they can do. So I pondered what I could do to help these people in my community in their struggle for a civil living in the United States. So that's when I also started doing more involvement for my school and my community. That's when I went to go protest and march to the Los Angeles City Hall when he was getting inaugurated. Um, and then finally, I chose to major in 2018 here at UC Merced with a focus on American politics and pre-law. So I was transferring my life from from LA to Merced to study here. And that is my presentation on the discipline of political science.
You were too hard on yourself. You did good.
Yeah, you did good, man.
Yeah, that was good.
Thank you. It was short, but I don't know more to talk about. Like, I think I just got it cleared off, like how the United States government started and what our three powers are. But yeah,
No, definitely. That was good.
Yeah, great job, guys. You all did a really good job.
Yeah.
After we end the call, I'll just try to upload it and I'll share it with you guys on the email. And if there's any problems or something, I'll let you guys know.
Sounds good.
Vikram, are you in the group chat for Snapchat or no?
Oh no, I'm not. Here, one second. You want me to add here?
Yeah, can you just write your Snapchat in the
Yeah.
chat and I'll add it. And I'm gonna stop the recording.
Okay. It's just this.